Organizers Jen and Aditi for the invitation, and sorry I missed the first few days of the workshop. I know there was one talk related to this, so there's going to be a lot of new jargons. Feel free to interrupt and ask, and hopefully there'll be new biology that you'll learn, if anything else, from this talk. So I'm going to tell you about some of the work from our group and more broadly from the field on this topic of using convolutional neural nets and their extensions to understand and predict properties of genomic DNA, and in particular non-coding genomic DNA. So just to set the stage, one key goal of modern biology and genomics is to build models that help us go from the genome, or genotype, to phenotype. And this is in particular relevant in the context of common diseases. So common diseases are almost all caused by a combination of genetic and environmental factors, and the combinations can be really complicated and complex. But even in those cases that the genetic contribution is relatively small, still genetics give a really unique perspective into causal mechanisms because it's upstream of everything. The direction of causality is controlled. There's no way for your phenotype to go and back in time and modify your genotype. So in a way, it's a very nice causal data set to understand disease mechanisms. So while this has been the goal for several decades, we're far from actually having such a model in a way that gives insight into disease biology. And the reason for that is that biology is complex. So between our genome and phenotype, we have many layers of gene regulation. So for example, we have our epigenome, transcriptome, proteome that we've talked a lot about, and many other layers downstream of that. And there's probably layers that are not in this picture because we haven't yet discovered them. Within each of these layers, we have different kinds of parts. So for example, in the epigenome, we have different modifications to the DNA, for example, different types of histone that modify the function of DNA sequences. In the transcriptome, we have different kinds of splice forms or different types of genes that are expressed, and so on. And then we have interactions between these parts within a layer. We have interactions across the layers and feedback across the layers. And on top of that, we have kind of environments loosely defined. This could be like the tissue environment or the cell type or environment of a person that modulates all these different interactions that are going on that connect our genotype to phenotype. So the grand challenge of computational biology, one way of framing it, is that to go from the picture that looks something like the top, where we can measure the parts with the different assays that now that can be readily applied, to a picture at the bottom, where we actually understand all the different interactions and feedback between and across the layers of biology. And really, to solve a disease, we really need access to the bottom picture, where we can say, this is kind of the space between our genotype and phenotype, and where can we intervene in that picture to most effectively address a particular disease mechanism. So in today's talk, I'm going to focus on our work and others that have been trying to solve the first part of this puzzle, mainly going from our genome to predict how the function of the genome gets unfolded across the two first layers of gene regulation, our epigenome and transcriptome. And in particular, there's this really exciting class of models that are now being developed that we and others have been referring to as sequence to function models. I think the term also gets used in the protein space. But the idea, and here I have a bunch of references, not just from us, but from many in the field, including Anshu's work that you heard some about on Monday. And the idea of these models, there are some variants of convolutional neural networks. They take as input genomic DNA across the genome, and they predict all sorts of functional properties that one can measure with a variety of assays. So for example, which transcription factors are binding, the chromatin accessibility of a given DNA input, gene expression of genes residing in that region, and so on. So for those of you that know the protein space really well, the computational framework or the problem is very similar in that we have some sequence as input and we're trying to predict stuff as output. But I would argue that this is actually a much harder problem to solve for several reasons. Two obvious one being that the stuff that we're trying to predict are actually not constant. They vary depending on the cell type and the context and the tissues that we're talking about. So it's many different modes of predictions that we have to make. And the second is that in the protein space, the input amino acid sequence, basically we can comfortably assume that that contains sufficient information about the structure of the protein. But here, there's uncertainty about the length of the input sequence that allow us to predict any of these entities because of long-range interactions in the genome. So regardless, this is an important problem that a lot of computational biologists are trying to solve nowadays. And generally, it's believed that if we can actually have accurate sequence to function models, we can address three fundamental questions in biology. First being to gain mechanistic insight into kind of so-called sequence grammar of gene regulation, and I'll maybe try to define that later. The second being, and this is kind of very interesting and exciting application of these models, is to perform in-silico mutagenesis. So the idea being is that if you have an accurate model, then we can ask it about arbitrary mutations in the DNA and see what are the functional consequences of those mutations are. And the third being, we can do some synthetic biology. For example, if we want to design a regulatory element that's only active in heart cells and no other cell types, we can use these models as Oracle, the same similar type of framework that we do in protein space. Okay, so in this talk, I'm going to tell you about three projects from my group that try to make advances in this sequence to function direction. So the first is our very early work in trying to use these models to understand what we call as the motif grammar of immune cell differentiation. And this is more just to set the stage to say what people know can be done well with these models. And in the second and the third part, I'll get to the more, I guess, cutting edge part of the science where we and others are trying to apply these models to understand differences in personal genomes and how that can translate to differences in their biology between individuals. And the second, kind of the shortcomings that we see with the current approaches and solutions to try to address those. Yeah.
Something that's happening here is like the different outputs, they are also, some are upstream and some are downstream.
Yeah.
Trying to predict everything from the sequence. Like, I'm wondering if there are models that also take into account the causal structure of the kind of intermediate risk or downstream outputs, or is that something that's
That's a really good point. So, these things, some of these are upstream of each other, but to a first approximation of all of these functional properties are encoded in genomic DNA itself. So in a way, you could pose it this way, and it simplifies the interpretation of the problem. But there are, there's things to be said for having more modular models where you try to predict one from the other in stages, and people do work from the modeling perspective, from using that perspective as well. Feel free to ask questions as I go on. All right, so maybe I'll go on with the first project. So this was got motivated to think about this problem based on a collaboration we have with a consortium called Immunological Genome, or ImGen Consortium. And it's basically a consortium of 20 or so immunological labs that generate really large immunogenomic data sets. So in this latest data generation effort, what they did was they isolated 90 different immune cell types from adult mice. So these are essentially all the different immune cell types that are functionally distinct and immunologists have been studying for 30 years. So the nodes in this dendrogram, each node is an immune cell type. Here is like the what I call the hematopoietic stem cells, so the immune stem cells, and they get differentiated and they become fully mature immune cell types. And an immunologist can isolate each of these cell types in intermediate steps and try to understand their differences. So the goal of this project was to try to figure out how biologically these cell types are different from each other in terms of this regulatory elements. So let me just unpack that a little bit. So they use an assay called the ATAC-seq assay, which essentially allows them to identify for each cell type the regions of non-coding regions of the DNA that are accessible to transcriptional machinery. So these are called the chromatin accessible regions, which I'm showing you here in different colors for different cell types. So all these different cell types have the same genome, but they have differences in terms of which genes are expressed. And one step upstream of that is which regions in the non-coding regions of the genome are regulating the expression of those genes that are expressed. So with this assay, they can isolate exactly which regions for each cell types are serving as some sort of regulatory element for that cell type. And the biological hypothesis is that, and I'm going to simplify this, but essentially the hypothesis is that if I were to study sequence composition in these regulatory elements that are exclusive to a particular cell type, I might discover a motif grammar, which is basically a combination of sequence motifs that transcription factor bind to and drive downstream gene expression for that cell type. So each of these cell types will have its own kind of combinations of sequence motifs which we can kind of visualize something like this, that determine which regions are accessible to transcriptional machinery, and that determines downstream gene expression. So that's like the high level idea of how we think these cell types are different from each other. And we want to use machine learning now to discover these differences in regulatory grammars that are encoded in genomic DNA from the different cell types. So to address this question, we pose two related computational questions. One is that on the basis of just like genomic DNA itself, can we predict the chromatin accessibility? So in which regions that region of the DNA and which cell type it's accessible? And the second is that if you have a model that can do this, then can we analyze it to understand what are the sequence grammar or these like kind of combinations of motifs that are deriving the predictions for each cell type? So this is now very standard in the field, but back in the day, kind of how we solve this problem, we just developed this convolutional neural network model that's a multitask model that takes as input just pieces of genomic DNA. So this is like the length of this genomic DNA is the length of the accessible region that comes out of the ATAC-seq assay. Then we can use one-hot encoding to represent it numerically, then feed it into a convolutional neural network. And as output, the model predicts, these bars represent the height of the chromatin accessibility in each of the cell types. So we have basically a continuous measure of accessibility of that region in each of our cell types, which we can then try to predict. So what we hope that this model learns is as follows. So what we hope is happening is that on the basis of the genomic DNA, the first layer convolutional neural network learns some sort of sequence motifs that are represented by the weights of the kernels that correspond to like our biological understanding of how chromatin accessibility actually unfolds, and that's based on the binding patterns of transcription factors. So we hope that these short sequence motifs actually represent short sequence patterns that transcription factors recognize and bind to, and then these motifs get combined in the later layer to create some sort of embedding of the sequences that is then predictive of chromatin accessibility in each of the cell types. Well, let me briefly show you two aspects of the result and how well this could work. So the first is like if you actually make these predictions and do cross-validation, how well can we predict chromatin accessibility just based on genomic DNA? So this is something that wasn't known before, and how well we can do this, especially at this cell type resolution that we have many related cell types to each other. So what this is showing is just one example of test sequence that the model hasn't seen. So for each test sequence, we're making a multitask prediction. We have the predicted chromatin accessibility, we have the ground truth. We can correlate those two sets of vectors to get a correlation value per input sequence. And this histogram here is now showing the distribution of the correlation values. So is this good or bad? One way to assess that is by creating some sort of null where we shuffle the label of of our outputs to figure out, what does the null look like. And with this null, we can say something like, from a statistical standpoint, about 60% of the test sequences we can make a statistically significant predictions for. So for those of you that apply machine learning a lot, cross-validation is often not end of the story, and we often need a harder, potentially out-of-distribution data set to really assess model generalizability. Yeah?
So when you say multitask learning, that's the number of cell types?
That's the number of cells. The tasks are the number of cells, yeah.
Can you speak to, how many cell types do you need to achieve some kind of performance or generalization power?
You can do this even for one cell type at a time. I think there is different views in the fields, and that if you use more tasks, you learn. It depends on the relationship between the cell types that you're using. If the cell types are very different from each other, then there's a lot of shared mechanisms that the model is going to be reusing. But in this case, okay, so I should have also put some numbers on it. So in what is the size of the training set? So in each cell type, you typically have around 100,000 regions that have chromatin accessibility. So in a sense, it's already, even for one cell type, you have a lot of data. And in this case, when we have data for across the 90 cell types, some of the regions are shared, so it's on the order of 500,000 to 1 million sequences where we have multiple labels for. Yeah.
I think I saw like a 200 base pair context window.
Yeah.
Does that matter much, or you have a sense of
Yeah.
how big you can go?
Yeah, so that's another really good question. So it depends on what you're trying to predict. So chromatin accessibility is a very local measure that's basically, I think most of the information is contained within that 200 base pair window. And we and others have done experiments to see if the window size impacts the prediction in that sense. It doesn't, but other things that you might want to predict, for example, gene expression that integrates information across multiple regulatory elements, that's where the size of the window matters, and you need much larger window sizes. All right. So are we sure that this, cross-validation tells us everything? One way to establish this is to go out of distribution. And in this experiment, so if you remember, I mentioned that this data was all from mice. It turns out that there was another big experiment done in human cell types where similar immune cell types were isolated and the same types of data was generated. And basically what we were able to show was that if you just applied a model that was trained on mice, it can still make sensible predictions on the human data set. I won't go through these plots, but essentially what I wanted to say is that this is actually a really hard problem to go between mouse and human regulatory elements, because if you do base pair by base pair alignment between regulatory elements in mice and human, only 10% of those regions might have like a base pair by base pair alignment across species. But we know that the immune system does have a lot of similarity between human and mouse, and a lot of the gene regulatory properties are conserved. And the conservation comes because the regulatory grammar, these combinations of motifs, are conserved, not the actual base pair by base pair alignments. So with this kind of analysis, we were able to show that the model actually has learned something sensible that can generalize across species. And in this case, that in one sense, you can say the data is very out of the distribution. So base to base alignment is very different. So yeah. 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 Yeah, sure. So I would say there's work from us and many other now that have really like grind us to a halt of like what are the differences in different model architectures. For this problem of chromatin accessibility, so our model has 3 convolutional layers, 2 fully connected layers. I think you can play around with 4 layers versus 5 layers and pretty much similar performance. And if you have much deeper models, you don't improve the performance. I think it's a simple enough problem that something out of the box that's not too simple will work. But there's a second type of predictions that I'll talk about in the second part that's where I think difference in model architecture makes a much bigger difference. But in this case, I think a reasonably medium-sized model tends to perform well, and you don't get a lot of gains from increasing complexity.
Labs or
yeah. So there's different ways to do this. In this context, so I showed you the result for cross-validation. In cross-validation, one thing that works well is to separate out chromosomes, because then you can guarantee that there is no overlap in the sequences that you'll see. So oftentimes people train their models on a random set of regions from a random set of chromosomes and then evaluate it on kind of unseen chromosomes. But that is good enough, and I think you can get pretty good sense of if the models are performing well. But I think if you want to be really sure, you need some out of distribution data that in this case, it made sense to go to a different species where we think there's some conservation of regulatory information. All right, so I think the second part of this first project that I wanted to talk about is like, it turns out, and you can say this was like lucky because the models weren't too complicated, but it turns out that the features that the models learned actually corresponded to a lot of known biology, which was amazing at the time because these are just like, they could learn any combinations of features and it doesn't really have to be biologically meaningful. So what do I mean exactly by that? So one way we could get a sense of that, and one very simplistic way, is to look at the first layer convolutional nodes and extract the sequence patterns that these nodes have learned. We're going to call those sequence motifs. And then look to see if they correspond to any biological known sequences of transcription factor binding motifs, which is kind of what would determine chromatin accessibility. So we did this with a two-step way. We extracted the motifs for each node using the weight matrices, and then we did an ablation analysis where we kind of nullify each node and measure its impact on the predictions. And so I'll just show you two results from here. So first of all, so each dot here corresponds to one a motif that was learned by by one of the filters. On the x-axis, I have the information content. So how different is that a learned pattern from random sequences? And on the y-axis is the influence using ablation. But I think what's the most interesting is the color of the nodes, which is whether or not that pattern that was learned matches to some transcription factor binding motif that was previously known in one of these big databases, cis-BP or JASPAR. And essentially, like what we observed was that majority of these motifs that are learned correspond to some transcription factor binding motif that is known to be important in the immune system. And based on this ablation analysis, we could go one step further and say for each of the cell types, which is on the columns here, so each row is one of the motifs that was learned, each column is a cell type, we can do this ablation analysis to see how important is each motif for each of the cell type. And in that way, we also recover a lot of known biology. So for example, there's two transcription factors, PAX5 and EBF1, that are known to be really important for B cells. So we see that in the model, it kind of comes out that way. So the color means the influence either up or down for that cell type. And there was a couple of other ones and some unknown kind of motifs that came up that were interesting. And we ended up further analyzing those experimentally. Yeah.
Are you taking any steps to ensure that each of these neurons in the first layer is learning kind of different motifs or orthogonally in some way? Because I would imagine, like, if you have, multiple nodes in that first layer learning similar, different pieces of information about the same feature than just single node ablation, or
Yeah, that's a really good question. So that's the reason we got lucky, because the architecture was simple enough and the dropout that we used was small enough. So I think if you had much larger networks, it wouldn't pan out like how it did when we did it with our just simplistic lens. But one step that we did take to kind of figure out how much redundant information is learned and whether ablation would actually be meaningful is to cluster basically the first layer nodes based on their activation patterns or the weight matrices, and then ablate them together in a group. And it wasn't that different from individual ablation of each of the kernels, I think because the model was simple and also because the signal in the data is very strong.
Because I know in things like some convolutional variational autoencoders, you can add some penalization term in some way such that you make it so all these nodes, you reduce the energy of activation across all the nodes in general, and you also try to make it so each individual node actually corresponds maybe to some orthogonal feature, or is it orthogonal as you like?
Yeah, definitely. And there's a lot of room for doing that because there's a lot of interest in physically interpreting the nodes that the models learn like this. All right, so kind of the last part of this first project is, so I was really curious in this question of how complex is the regulatory code? And what I mean by this is that how many motifs are needed to make a prediction, a good prediction for an input sequence? And essentially, we answer that through ablation. But what we saw is that there's wide variation in the number of sequence motifs that the model is using to make prediction depending on the input sequence. So this distribution shows for a given input sequence how many of these motifs are used to make the prediction that the model does. You can see for some input sequences, on average, the model is using one or two motifs, and for some others, it's using a much larger number of sequence motifs to make the predictions. So I'll go to the second part of the talk, which is more recent work on using kind of models, like I mentioned, but more complicated and more modern versions of them to actually understand differences in individuals' genome. But let me set the stage for that. So I told you about one model that we developed, AITAC. That was early days. But since then, there's been a lot of development in this field, and there's a lot of very state-of-the-art, very large models now for predicting a variety of functional elements. Most of them are very multimodal, so they try to predict many different things from genomic DNA. So some of the names of these big models include like Informer, Basenji, Big RNA, Borzoi. I don't know if any of you have heard of any of those, but those are the state of the art for these kind of sequence to function modeling. And all of them basically have the same training recipe, as I call it. And the training recipe is that regions in the genome are treated as training examples. So we walk across a single reference genome where we can measure a bunch of stuff from, and then we take windows across the genome, and so we have the sequences that lie at that window, and then we can measure a bunch of stuff that coincides with that window, and then we predict that bunch of stuff from it. So we can do this in one cell type at a time or multiple cell types. The state-of-the-art model nowadays do this for many, many different cell types where we have measurements for. And in particular, kind of the most immediate type of output that we want to be, as in the field wants to predict, is gene expression, because that's the output closest to the phenotype. And all these other things that we can measure are like proxy or a way to soft regularize the model so that be able to predict gene expression well. And I would say the other thing that I would say is like the state-of-the-art models today for predicting gene expression, the window, the input sequence window that they take is very long. It's about, I think, like 200,000 base pair of input sequence, and on using that, predicting gene expression for genes that kind of coincide in that window. So this is basically the training recipe that all these models are using. We're windowing the genome, and then we're predicting stuff from it. Consistent with this training recipe, the performance is measured based on leaving out some regions or some chromosomes, and then checking to see if you can make accurate predictions for regions that were left out. So in the case of gene expression, and I'm going to particularly talk about this model called Informer, which is one of the state-of-the-arts here. So this is an evaluation that we did independent of the Informer paper, but many other papers have shown this now, is can leave out some of the chromosomes and then ask the Informer to make predictions about genes that coincide on those left out chromosome. And you typically see like really robust, good predictions that, so this is predicting gene expression in the brain, which is one of the tracks that the model is trained for. And you can see predicted versus observed, there's high correlation. So the model can somehow distinguish which genes have low versus which genes have high correlation based on the sequence composition of the genomic DNA centered at that gene. Yeah. Yeah.
So can you, like, I wish Anshul were here, because I could swear Anshul said that we can beat those models using stuff from like, 10, 15 years ago.
Yeah, so it depends on what exactly you're predicting. So Informer is predicting many things. One of the things is gene expression. Other things are like chromatin accessibility or things that are more localized. For more localized predictions, you don't need a really big window. For gene expression prediction, in theory, because enhancers could be so far from the gene, you might need a big window. Although there has been papers that shown in practice, you actually don't need such a huge window because the model is not really utilizing that very long, far away sequence information. I think there is biological information in those, but maybe there's not enough training data to learn that information coherently, and that's why you can beat them.
Is your point that it does well, or that it's state of the art?
It's state of the art, and it does well in this particular task. And I'm now going to tell you what people care, like what actually people want to use. This is not the task that anybody cares about. This is a proxy task for saying, I learned to predict gene expression. But the task that everyone cares about, and that's like the motivation behind these models. Yes, Hunter?
I was just curious for predicting gene expression in general, how relevant is the gene itself? Like, would it be helpful to know what the gene is, like with the
Yeah, so that brings up a good point, that these models generalize across genes. So you actually don't know what the gene is because your training examples are just sequences. And the point is that within like basically the information about how much a gene is expressed is contained within some distance of that gene, like some local distance of that gene. And that's what the information that the models are exploiting.
So just so with the, say, 30,000 order of genes, how many sequences are included as
Yeah, so that's a great question. So that goes back to like, what's the training size here? So because when you're predicting gene expression, you're limited. There's an upper bound of number of genes that you have, and that's the upper bound of the number of training data points that you have. You can expand that by having measurements for many different cell types. So then you can multiply your number of genes by the number of cell types, and that would be the number of training examples. But in this training recipe, and I'm going to now tell you an alternative in like a few slides, but in this training recipe, you're bounded by the number of genes that you have.
And then multitask across different cell types.
Okay, so this was like, and consistent with the way that these models are trained, if we leave out some regions of the genome and make predictions about gene expression, they work well. But this is a proxy task. What people really care about is using these models for in-silico mutagenesis prediction. What does that mean? The idea is that if you have like a trained model that's behaving well, what we can test it on is I can input an arbitrary sequence, and then I can mutate any arbitrary base to a different base and ask the model to contrast those predictions to see what is the functional consequence of variation or mutations in the genome. And if we can answer this question, this is like will have hugely impact in understanding when genetic differences between individuals will lead to differences downstream in their biology and predispose them to disease. So the end goal is to be able to do some sort of what people refer to as ISM map, where for any given arbitrary input sequence, I can mutate every base pair or every combination of base pair and kind of predict what would be the impact of that kind of ablation or mutation on the functional properties that I care about. And so it's widely believed that like those models might be able to solve this problem, and I'll show evidence in a second and why so.
Mutations.
Yeah.
And I'm not a biologist, but I can imagine that most mutations are extremely deleterious.
Most mutations are not deleterious at all. There is like many, many differences between our genome. A subset of them are actually functionally do anything because there's random mutations everywhere. And if it was deleterious, we would not be born. So
I'm just trying to understand the nature of extrapolation off this manifold.
Yeah.
Is there some regularization where it's like, if it's off the manifold, I don't think it does anything, or I don't know.
Yeah. Okay. So I will show empirical result of what happens, and then I'll tell you. But I think you're on the right track. It's not exactly an out of distribution problem the way it's, I guess, conceived in other parts of machine learning literature, in the sense that the difference between these two sequences is very minor, but we don't know exactly when the single variation would actually cause a difference. And I think it boils down to being a resolution problem rather than an out of distribution problem. Okay, so I guess when biologists see this, their first reaction would be like, why would we expect a model to actually predict the effect of genetic variation effect? There's three reasons. One, conceptual. So the conceptual reason is that if there's really a sequence grammar of gene expression, like I showed you before, and the model is using that sequence grammar, then it should be able to predict if the mutation is impacting that grammar, then it should already know that it's going to result in some sort of functional consequence. And if it's not impacting that grammar, then it's not, it doesn't have a functional consequence. But then there's two empirical lines of evidence. One is through matching up these kind of ISM maps with experimental mutagenesis experiments. So this is from one Informer paper that was published 2 years ago. And what they showed was that, so this is experimental mutagenesis of an input sequence where you experimentally mutate every base pair, and the height of that bar is like how much effect that mutation had on gene expression. And you can make predictions with Informer in silico. And then the idea is like for several regions where this data was available, there's good correlation between experimental mutagenesis and in silico mutagenesis. So that's one line of evidence. The second line of evidence is, it's a little bit involved, so I'll like skip some of the details, but the idea is that this is actually a very historical question that people have been asking in population genetics to understand the effect of genetic variation across individuals. And the way this is done in population genetics is that you for a bunch of individuals, you collect their genotype information, so their genomes, and then for the same set of individuals, you have their gene expression information, and then you can correlate pairs of features, one from the genotype space, one from the gene expression space across individuals to tell which base pairs across the genome correlate with differences in gene expression across individuals. So you don't need to know all the details on this, but the idea is that you can do this. You can answer this question with population genetics, where you can generate genomes and gene expression. But it's a little bit more complicated to do it this way, or a little bit difficult to interpret to do it this way, because here you also have the problem of confounding. So the idea is that in the genotype space, regions in the genome that are close to each other, genetic variation correlates, so that you have this concept called linkage disequilibrium, or LD, which just means your input features are very correlated depending on their location. And because of that, you can't really tell which one is the causal one driving the result. So to use this data, the second step that you need to apply is statistical fine mapping approaches that people have come up with to try to disentangle of a set of a bunch of correlated SNPs or variants, which one is the one that's more likely to be causal. So there's a second inference step that people can use here to isolate the regions or the base pair positions in the genome that are likely to be causal to gene expression. So if you take this population genetic data and you ask kind of one of these S2F models, including Informer, to distinguish the causal genetic variants from the ones that are not causal, these models tend to do okay. So this is an example of a plot from Borzoi, which is another state-of-the-art model. And what they showed was that for genetic variants where we know are causal, the model can actually distinguish those from the non-causal variant. And that's essentially what that figure is summarizing. Yeah.
Generally better than Informer?
So Borzoi is a sequel to Informer that's supposed to be better. I guess it's like, yeah, it's
like and you think it is?
It is, but not in this task. It is, but I don't think it has substantially solved the problem in a way that it'll just come in like one slide.
And is it easy to, like, in one sentence, describe the advancement in
Borzoi as using more data modalities and then using them in a more refined way in terms of not requiring a lot of pre-processing on the data side.
the same context size?
I think that, yeah, the context side is the same. And I think better model architecture. Okay, so we had a simpler idea of how to, so basically the idea is like we want to use these models to be able to predict differences in gene expression between individuals. And these two proxy evaluations that I just showed you with experimental mutagenesis and these kind of like population genetic approach are each have their own issues that I try to a little bit allude to. But we had a simpler idea that bypasses the issues with all those evaluation. And the idea was very simple. So nowadays there's a bunch of cohort studies where people measure whole genome sequencing. So we actually measure for every person their entire DNA sequence as a string. And for the same set of individuals, people generate data modalities like gene expression. So there was one cohort study that we're working with, it's called ROSMAP. It has such data for about 800 individuals. So our idea was like, why don't we just construct these personal genomic DNA sequences, feed it into a model, and check its prediction and compare it to what's actually measured as opposed to going to these other proxy metrics. So I'm going to first tell you about one example gene when the models like this actually performed really well, and then tell you what happens when we try to generalize to the entire genome. So here's one example gene. And I should say like the results that I'll show is specifically for predicting gene expression and specifically for this one model Informer. But in our paper, we show that this is a common result that you would see on any of the existing model of today that are using the same training recipe. And the existing models are all using the same training recipes. Okay, so first let me tell you an example of one gene where this works really beautifully, which is this one gene called DDX11. It's kind of like a positive control experiment. And the reason for that is it's a really easy gene. So the gene expression between individuals for this gene varies a lot. So some people have very low expression, some people have very high expression. And that difference in gene expression can be explained by a single base pair variation in their genome, so a genetic variant that's known to be causal. And if you stratify individuals in any data set where you have gene expression from the brain, you can see that the individuals that have like the CC allele have lower expression, the individuals that have AA allele have higher expression. So it's a very clean gene in terms of there's large effect from one single genetic variant. Now, in this case, if we create these like personal genomes and feed them into a model like Informer, It's actually kind of difficult task because these personal genomes, each pairs of individuals might have up to like 2,000 or more differences in single variation across their kind of the genome that's input to informer, but only one of those, exactly one, is deriving the differences between between individual in terms of gene expression. So we can input the sequences to the model, and for each individual get a prediction of the model's prediction of gene expression for that person versus the observed. And in this case of EZ gene, we see there's like really good correlation, so correlation of 0.85 between observed and predicted across all the individuals. And it's actually very nice now if we could do this systematically, because if you were to use the model that I mentioned and perform in-silico mutagenesis to figure out what is the exact genetic variant that's driving the result, so that's shown by these two plots here. So each dot here is like the ISM value applied to one of the observed variants in the population. We see that there's exactly one variant that the model is relying on to predict high versus low versus medium expression. Then we can look further within here to see what's happening, and there's some really nice biology. What's happening is that in the reference allele, that region is like doesn't impact gene expression, but in the alternative allele, there's an insertion of an A instead of a T, and that results in a repressive motif that reduces gene expression. So there's like a nice biological explanation of why the model makes the prediction. Is consistent with this idea that the model has learned the sequence grammar that determines high versus low gene expression. So this was easy case. What happens if you do this systematically? So it turns out that we can do this for every gene and look at the results. So now each dot represents the correlation between observed and predicted for each gene across individuals. That's the informer on the y-axis. And on the x-axis is a very important baseline measure, which is, I'm not going to go into the details, but it's a model called PredictScan, which is basically a population genetic model just to estimate a lower bound on how well we can predict gene expression from genotype information. So it's really a baseline for saying, for a given gene, how much genetic do we think affects this gene expression? And basically 2 observations came out of here that motivated kind of ongoing work that I'm going to tell you in a second. But the first observation was that the direction of the correlation seems to be almost random, even for very significantly predicted genes. So there's a class of genes here that the model is predicting the genetic variation to reduce expression, but it's really decreasing expression or vice versa. So that's why you have this like set of genes with highly negative correlated values between observed and predicted. And the second observation was that even compared to like a baseline model of PredictScan, this kind of complicated S2F models seem to be underperforming, which was not expected in the field. So people thought, now we have this fancy machine learning models, we're going to be doing better than what people have been doing in population genetics. But think about it, it's not that surprising based on how these models are trained. And I should also mention that, so I didn't mean to say like Informer is bad. Informer is a state-of-the-art model, but all the other models have the same issues in terms of direction of science and underperforming because they're using the same training recipe. And what we think is happening is basically the space of sequence grammar is very complicated. And when we use these models, when they use only the reference genome to predict high versus low expression across genomic locations, they learn a subset of regulatory grammar that's important for determining functional consequence of genetic variation. So what this figure is a little bit involved, what this is showing, but showing is that this is often what we observe, is that if we know what the causal variant is that's resulting in high or low expression across individuals, that tends to fall outside of regions in the genome that the models are actually using to make predictions. So they didn't really learn a motif grammar that encompasses these like causal variants that impact inter-individual variation in gene expression. Yeah.
On the last slide, is this PredictScan is like a kind of ETL approach?
Yeah.
So like in what sense is that actually a ground truth? Like how can you benchmark against that?
It's not a ground truth. It's more like a baseline in terms of telling you how well can you even predict gene expression from genetic information. So for some genes,
but it may be predicting it wrong, right? PredictScan itself may be predicting it wrong.
Well, okay, so but the sign of the correlation is like, that's like if the sign of the correlation with the observe is negative in Informer and PredictScan doesn't have the same issue, I think that's like pretty conclusive that PredictScan got the design correct because it's consistent with the observed data. I think it's like just an underestimation of how well you should expect to predict gene expression of each gene based on genetic information.
I'll ask you more after.
Yeah. All right, so is there any more questions on this slide? Okay, so just a few more slides left, and I know people are hungry, so we'll get through them together. All right, so I guess jumping to the very high level, the issue, which maybe like you all already figured out, is that the training data is too sparse. The training data in terms of the diversity of the sequences that the models are seeing. Like really, if you use just a single reference genome to train these models, at the very most, we have 30,000 sequences that are centered at gene's transcription start site, and that space of sequence diversity is not sufficient to be able to predict inter-individual differences in genetic variation. So even if you add one extra genome to this kind of training recipe, we already increase the number of training examples by number of genes because each individual can contribute all their, the sequences around all their genes to the model. So I guess a simple solution is like just train the model on larger data sets. But this is my favorite cartoon, and it turns out that it wasn't true. So the guy says, what is your machine learning model? And he says, I have a bunch of linear algebra, you pour data, and then you mix and mix, and the good things come out at the end. In this case, it didn't come out at the end. And that was for two reasons. One is kind of scalability. That's actually training these models very expensive, and I'll show on the next slide how expensive. Maybe, I don't know what is it in protein space, but pretty expensive in our space. The second, and so we need to like figure out how to do that effectively and efficiently. And the second is, it turns out that there's two types of variation in the data that we care about. There's variation in gene expression from gene to gene, which are very large. So some genes are like really highly expressed, and some genes are very lowly expressed. That could be orders of magnitudes different between the expression level across genes. And then there's the variation across individuals. So for the same gene, some individuals are expressing that genes higher, and some individuals are expressing that gene lower. But that variation is much more subtle. It's not orders of magnitude. It's very subtle. So we care about both of these variations. And if we don't force the model to focus on the more subtle variation, we just end up learning these like larger variation across individuals. So what is the scalability issue? So like if you just have one reference genome, like Informer model, with one GPU, it takes four days to train it, but then we just have to parallelize. If we're down to like 32 GPUs, we can do it in an hour. So if you want to train it on many, many genomes, we need many, many more GPUs. But that's just a problem of like parallel algorithms. Second problem is more conceptual. And I think that's a very neat way to solve these across machine learning problems where you have subtle effects that you want the model to learn. So the idea was to use this contrastive learning design where you have this model, it's a convolutional neural net that takes as input pairs of sequences and then contrasts them. So for a given gene, we have the reference genome at that gene, and we have an alternative sequence, which is an individual's genome at that gene. And then we predict as output the average gene expression for that gene across the individuals, plus kind of the delta for the individual. So the difference is for that individual to the reference and this, which just helps. So the idea is that if we break out our loss in that way, then we can really force the model to learn the more subtle variation between individuals, not just kind of the variation from gene to gene. And just two results slide. And this is preliminary unpublished work. We were able to train this model on multiple genome, and the big cohort that I mentioned has 800 individuals, but we only did it for 100 genes for computational reasons. But even when we have 100 genes, we basically have 100 by 800, because I have 800 individuals training examples. So what we could show was that at least now the correlation between observed and predicted per gene is matching kind of the baseline that we thought we should at least be able to match. And this is much better than kind of Informer and a simple CNN model that that doesn't really get the sign of the correlation correctly even. And you can see, and well, the question you might ask is that if all you did was like match this PredictScan model, which people have been using for a long time, what's the point? I guess the point is that when you have a mechanistic model, then you can ask more refined questions and get more causal insights from the model. So what do I mean by this? So for example, there's one gene that Informer couldn't solve, but now we can predict gene expression across individuals based on there's differences in their genome, and we can precisely pinpoint why are the individual have differences in gene expression. It turns out that there's one particular mutation or variation at one location in kind of near this gene where insertion of that alternative base results in disruption of repressive motif that then increases gene expression in the alternative individuals. And this is just the last slide saying that this is not just like cherry picked and one gene, but we can kind of do the same analysis for the other 99 genes where we actually know the causal variant from previous experiments. And we can show that even though we didn't tell the model which one is the causal variant, it can distinguish the causal variants from the ones that are in LD but not causal. Okay, so I'll end with 3 conclusions, I guess. One is very familiar to this crowd. Not all evaluation and training strategies are the same. And depending on exactly how you evaluate your model, even on the same data, you may come up with different conclusions. Other more substantial ones, I think sequence-to-function models are a really exciting area of development and understanding how the function of genome unfolds at these different layers of gene regulation. But I think there's some there's ways to go to be able to actually make these models accurate enough to be able to apply them and understand differences in person's genome that results in differences in their biology. So with that, thanks to the students that did this work, in particular, Shiming, Anna, Alex, and Elon for the second and third part of the talk. Collaborators Maria Chiquina, Naila Ioannidis, who is here and we've been talking to a lot, and kind of the MGen consortium that got us started in this direction. And if there's time, I'm happy to take more questions.
I'm going to ask the first question. In this last part with the contrastive loss, you have like 3 different parts of the loss function. Yeah. So like, do you just scan hyperparameters to reweight them? How important was it? And what's the upshot of such an investigation?
Yeah. So I think that this is the hyperparameter on this one is the most important for getting the subtle effect of differences from that individual to their reference. That makes a big difference. And then having these 2 is a little bit redundant, but just empirically, we see that having both of them somehow helps during training. And it's like there's no theory that I could explain why, because they're redundant information.
Do you remember the value of lambda you pulled out?
No, but I can get you that number in an hour.
Okay.
Hi. So in cases where, let's say I have a cell atlas and I know that given a certain diagnosis of my patients that they have a log-fold change of X for, let's say, some random gene, how possible is it using this model to kind of take that information and then try to generate a set of candidate mutations that might actually be influential to kind of generating the effect that you actually see in have you tried that?
So I guess can I rephrase the question? So if you already know there's a patient and there is a log-fold change of X between their expression of an important gene and the reference, and you want to figure out which of the variants are deriving this, that's the exact application of models like this. And I think if you can solve that, you've solved a really big question in biology. So I guess in some cases you can already do this, and I showed you in this case of like DDX11, which was this like really easy gene, you can already do this really well. So this would be the example of your patient that your patient a set of patient might be here that have higher expression of that gene, and the model figured out that they should have higher expression because the model identified this one genetic variant that the references don't have it, and then the patients have it, and that and then the model can tell you that's what's happening. That's why some people have lower and some people have higher expression. But in general, I guess if it was an arbitrary gene, arbitrary across the genome, I think we're still far from doing that. And it's very active research to figure out exactly how to make the models better. And I don't want to say we're far from that. There is a lot of hope that we're getting closer to that because there are cases like this that can already be solved. Yeah, like if you gave me an arbitrary gene, I would have to know more about genetic diversity in that region across individuals to tell you, like, is it going to work out or not?
Gotcha. And then just a small thought. So it works for like individual point mutations. Have you tried also looking at alternative splicing patterns? Because there are a lot of genes there, like you'll have like a lot of bases just change.
Yeah, we haven't looked at that splicing in general with these models, but there's a lot of work that does that. And I think that generally you can do that well, because maybe better than you can do pure gene expression, because the grammar of splicing is more confined in some ways.
Gotcha. Thank you.
There was 2 questions. Oh, maybe Bear was first. Sorry.
Thanks. I have a clarification question on the loss. I was trying to think about why the individual, the redundant input might help.
Oh, sorry. Here.
So is R hat just the delta?
Okay, so this might not exactly correspond to my picture, but R hat, let me see how I had it in my notation. So one of them is like the mean, I think the hat is like, doesn't matter, but like one of them is like the reference. This is for the cross genes, and I guess this is for R hat is, one of them is the reference and one of them is like the individual. So this doesn't exactly correspond to this. I think there needs to be another term for it to make sense.
I see. So the reference comes into the loss for every individual.
Yeah, it does. So it's redundant information in that way too. And I guess another way to do it is basically you contrast every pair of individual with each other. I think it will give you similar results. This way you have a lot of redundant information, but you do less contrast.
And you said you found this helped compared to just using the reference and the delta as output? Or
Yeah.
Okay.
There's 2, and then there's you. And Jennifer can put a stop whenever you want.
Thanks. I'm looking forward to the deep allele paper. It seems really cool. I have a question about how you think about these sequence motifs in general. So throughout the talk, you mostly talked about them acting on a single gene, but to what extent are the sequence motifs themselves convolutions of other sequence motifs, and can you use models like this to deconvolve them and figure out contributions like inputs and outputs?
Okay, so by that, do you mean like a given sequence motif can actually be composed of 2 smaller units?
2 smaller units, or a merger of two other motifs, so it's acted on by 2 transcription factors, or it will act on more than one downstream gene?
Yeah, I mean, that's a really good point, because biology is very complex and we actually don't understand what the words are. Like in some sense, like I don't know, it's like we're trying to rediscover how biology actually works so we can make those definitions. But I guess in how I was thinking about it in the context of this talk, and especially the earlier part of the talk, is that they're based on transcription factor binding motifs that we could chip for. So they correspond to individual transcription factor binding motifs. But in another sense, you can say a combination that always co-occurs, and that's just one unit on its own. But yeah, it's complicated like that.
I was wondering how much CRISPR and gene editing is changing your ability to like just do the causal interventions and generate that type of data or test hypotheses.
That's a really good point. So I think so far the way people have been using it in terms of model evaluation and not so much using the data to train or fine-tune the models, but I think that's another direction that can be pursued once those data become larger, and that you can combine them with like natural genetic sequences to kind of better delineate causal signals.
I really enjoyed the talk. I have a question on the contrastive part. So per my understanding, there are really like 2 problems you're trying to tackle. One of them is obviously data sparsity. Like you really want to, I guess, pick up the parts that are very subtle, but really are causal. And I feel like the contrastive really tackles the difference parts really well because you subtract out the background. But I feel like this doesn't necessarily encode a bit of the causal, I guess, flavor into it. But because in theory, like the correlations can still be learned without necessarily having to be causal for the downstream task, which I think is still a big improvement from the from the previous approach. But I'm curious on whether you've thought about like ways to encode some of the other modalities into this, like the attack-seq or I guess other RNA-seq data, which you have shown to be more, I guess, a bit better to capture these causal modalities into this work as well.
Yeah, so that's a really good point. And I would say like eventually what I envision is like models like this become very multimodal, that you're predicting many different types of information, and that helps to regularize or implicitly regularize the sequence space. But yeah, I guess that's the end goal, but we're doing baby steps one at a time. There's one question there and then one there.
They yell without the microphone. You can hear me, right?
No, it's not about that. It's for the online recording.
Thank you. I wanted to follow up on your comment regarding the scalability of these models. One would think that the number of SNPs in the human genome is finite, representing the population as a whole. And I was wondering whether you thought of how can the model architecture perhaps be adjusted to really kind of reflect the fact that really the number of SNPs in the coding region or non-coding region you're looking at is X and not all possible permutations. And whether this could be somehow used to
I mean, I guess it would be like, yeah, different representation of the same data, although it's much larger than I would have thought a few years ago. So basically, if you do whole genome sequencing between two random individuals at just like single base pair level, I think there's like 20 million differences. And if you consider many, many different individuals, then that number starts to scale up because there's a lot of rare variation between individuals. So I think you still see a lot of genetic diversity. It's just, but the positive data, I would say, is very sparse, and that most of the variation don't do anything. So you have a lot of negative examples of variation that are not functional, and you don't know which ones are the ones that are actually functional in some way. But I think it's a good idea. Like there are ways that you can encode into the model, like regions that just never change based on conservation, or things that never vary in populations versus things that might change.
Or you can just look at regulatory elements that can change gene expression based on your current model, for example.
Yeah, I think that definitely like room for development and thinking in that space. Architectures need to be changed if we're really thinking about these like multimodal across many layers of gene regulation with like many genomes, then scalability will become a real issue.
I think in the interest of time, we need to close it up here. So let's just thank Sara again.